we had discussed the point up to the primary and secondary response. I have to tell you about two more important points regarding the B cell activation. One, I have already mentioned about class switching, which is mentioned here, where the B lymphocyte switches from production of only IgM to produce other antibodies. That is the key feature of the B lymphocytes, and that would happen when T cell dependent B cell activation takes place. Okay. In addition to that, two more other things that happen in the B lymphocyte. One very important process will be allelic exclusion. Please remember these three key features: class switching, allelic exclusion. Allele means gene, allelic exclusion, then somatic hypermutation. All these 3 points I'll be explaining to you in detail when I'm going to talk about the antibody genes. Okay, antibody genes, organization of the antibody genes, and how the antibody is going to be produced with the combination of the recombination of variable region gene with the constant region gene. Okay, so just to tell you briefly, as I've already explained to you about class switching, what is meant by allelic exclusion? You all know from genetics that a given gene will exist in pairs. If it exists in more than one pair, it will be multiple alleles, right? So, for example, IgG gene has got a pair of genes. Similarly, IgA, like that. So, one gene is inherited from the father, one gene is inherited from the mother. Okay, the number of the variable regions and constant regions meant for this IgG gene will vary from paternal and maternal. Okay, now when that gene is going to recombine, the constant region gene is going to recombine with the variable region gene. It can choose any 300 of the variable region genes and combine with it. At that time, as I had mentioned to you, it can produce a productive B cell receptor or non-productive. When I say productive, that will be high affinity or low affinity receptor. Non-productive means it will not show any affinity to the antigen. So by trial and error, B lymphocytes will be trying to have the best fit receptor. In that process, one allele, depending on which receptor is going to be the best fit, okay, that has high affinity for the foreign antigen, that will be chosen, and the other allele will be silenced. In the sense, other allele will remain excluded. One allele will be selected. That is what is meant by allelic exclusion. I will explain this phenomenon with diagrammatic presentation when I'm going to talk about organization of the genes, immunoglobulin genes. Then coming about somatic hypermutation. Immediately after this part, I'll be talking about the antibodies. That time you will come to know that the specificity of the antibody, that is affinity and avidity of the antibody with the antigen, lies in its FAB arm, where the variable region is present. And that variable region has domains of amino acids, and we have seen those amino acids will establish the bond with the antigen either by positive negative interactions, ionic interactions, hydrogen bond, weak van der Waals force. Now, if it has to show affinity and high specific affinity, that specificity will be there in the variable region. And that could be achieved only by the mutation of the variable region. And that mutation is taking place in the somatic cell. And the somatic cell is the B lymphocyte. And tremendous amount of recombination and mutation will be taking place. How it takes place, when I explain about the genes, you will come to know. And that somatic mutation is called as somatic hypermutation. And it is only through that hypermutation, few of those mutations will be productive. Therefore, the receptor will be productive with high affinity and low affinity, and few of the mutations will be useless. Why it becomes useless, that also we will see in the diagrammatic presentation. For the time being, keep these three different phenomena that take place in the B cell activation in mind. And if the explanation is asked in the organization of the immunoglobulin gene, how does the class switching mechanism take place? There you can explain it in a different way with this diagrammatic sketch. But here you can just give the explanation in plain simple words that class switching is nothing but the switching over of production of antibody of IgM type to other antibody types like IgG, IgA or IgE, which happens when the B lymphocyte commits itself for the foreign antigen. Okay. Similarly, somatic hypermutation is a phenomenon which happens in the variable region of the antibody region to put in the best fit receptor for the antigen interaction. And in that process, some of the mutations will be useless and some will be low productive, that is low affinity, and some will be high affinity. And those are chosen to have the receptors on the B lymphocyte. And allelic exclusion, where one of the allele, either the paternal or maternal, will be chosen for to make the B cell receptor, and that allele which is chosen to make the B cell receptor will become the selected gene for the production of the antibodies. Whichever the receptor that comes on the surface of the B lymphocyte, that gene will be the final gene for the antibody production in the B lymphocyte with the best fit receptor, and that will happen only with the T cell dependent B cell activation. Okay, so that is as far as the B cell is concerned. Now, this is the comparative table. You can refer to it for your information. I've already mentioned that the primary response will always take longer time than the secondary response. Secondary response is also called as booster response, in the sense already the B lymphocytes are conditioned for the antigen, and they have to just respond immediately through the memory B cells. You may have a look at it on your own, and there is nothing much to talk about. Now I will talk about little bit about the B cell receptor. We had spoken about B cell receptor repeatedly, and this is the monomer, the large molecule that is shown as MIG, that is membrane bound IgM. It is supposed to be M molecule, IgM molecule, and that membrane bound IgM molecule has two FAB sites for binding of the antigen, and it is a monomer. And look at the cytoplasmic domain of it. The cytoplasmic tail is very, very small. It is hardly about 3 to 5 amino acid in length, and transmembrane domain is about 40 amino acids in length, and there is extracellular domain, that is this domain which is just projecting out of the plasma membrane, is about 26 amino acids in length. Even though it is a macromolecule which has the ability to bind to the antigen, and we had seen that antigen binding in the first diagram when we had spoken about, see here the binding of the antigen takes place to the FAB region, all right, FAB region. So when the antigen is bound there, this molecule is supposed to facilitate signal transduction, means activate the B lymphocyte. As soon as the antigen binds in the FAB region, it is supposed to bring about changes in the cytoplasmic tail region, where phosphorylation should take place and signal transduction to switch on the genes for the various processes. But unfortunately, because IgM molecule has a very short cytoplasmic domain, that is 3 to 5 amino acid, it fails to do the signal transduction. That is why each IgM molecule on either side, it has not been shown this side, but on either side is accompanied by these two subsidiary receptors, Ig alpha and Ig beta. Okay, this is called as heterodimer. Ig alpha and Ig beta is called as a heterodimeric receptor, and that Ig alpha Ig beta receptor, which is present on either side of the IgM molecule, when the antigen binds to the FAB site, it will help in recognition of the antigen because of the bending of this arm, and that arm will have established contact with this region, and the antigen antibody complex will be formed between Ig alpha, Ig beta plus the FAB arm of the MIGM. The arm will bend down after the antigen binds here. Therefore, it establishes simultaneous contact with Ig alpha and Ig beta. Similarly, it happens on this side. Now you know, because these two molecules, Ig alpha and Ig beta, are provided with long cytoplasmic tail. See, Ig alpha is being provided with 61 amino acid residues, and Ig beta is provided with 48 amino acid residues. That's quite a good long cytoplasmic domain. Minimum length of the cytoplasmic domain required for the signal transduction is at least about 25 amino acid residues, which is there in IgG molecule. IgG molecule has 26 amino acid residues in the cytoplasmic domain, but IgM does not have it. So therefore, these two co-receptors, which are subsidiary receptor, co-receptors, facilitate for the signal transduction. All right, and because of these receptors, the signal transduction could take place. That is why in this diagram, in the diagram that was shown here, they have shown here signal 1 from here, this area, indicating that the Ig alpha and Ig beta are present here adjacent to the membrane bound IgM, and that signal transduction is taking place from there. All right, it is taking place from there, not from the IgM by itself, because it doesn't have that cytoplasmic domain. So therefore, they have shown the position of that signal transduction there. So the blocking of this receptor, let us say, whether to prove that this receptor is very, very important, it is not just only IgM is sufficient. Even though the antigen binds here, suppose if you block this receptor, Ig alpha and Ig beta, with a monoclonal antibody against it, the monoclonal antibody will come and bind, and it will block this receptor. Then what happens? If the the membrane bound IgM binds to the antigen, then that B lymphocyte will not be activated. That is one of the approach that is taken, all right, to immunotherapy. Like suppose you want to block a B lymphocyte that is getting activated and showing autoimmune response and producing the antibodies against one of your internal antigen and causing damage to it, you can block that B lymphocyte using a monoclonal antibody that blocks this Ig alpha, Ig beta receptor. It will block on both sides. Even though it is shown on one side here, it will block on both sides. Then the signal transduction does not take place, so B cell will not get activated. Other approach will be just block the FAB arm of the MIGM. FAB region of the MIGM is blocked by the monoclonal antibody. Then it will not be able to bind to the antigen. Okay, so if it doesn't bind to the antigen, Ig alpha and Ig beta on their own cannot bind to the antigen. That is also there. They need the assistance from IgM. Okay, they need the assistance of the IgM, that the antigen has to be bound to the FAB arm of the IgM. Then only it can communicate with this by bending, and then Ig alpha, Ig beta will succeed in signal transduction. So both ways they are interdependent. Both ways they are interdependent. MIGM is dependent on Ig alpha, Ig beta, and Ig alpha, Ig beta is dependent on MIGM. They can do the signal transduction only after the FAB arm of the MIGM receives the antigen. There are many other receptors which are very important, but I don't expect you to remember these receptors as such. See, this C3D receptor that is shown is the complement receptor. Okay, C3D receptor is the complement receptor. And that complement receptor that is bound to the antigen can communicate to the MIGM FAB arm. Similarly, there is CD19, CD21. CD19, CD21 are very important receptors and they are overexpressed in B cell lymphoma. B cell lymphoma, that is the cancer of the B lymphocyte. Okay, and these two receptors are generally targeted by monoclonal antibodies to control that cancer. CD19 and CD21 receptors are blocked by the monoclonal antibody. They are also B cell co-receptors. Okay, we'll not bother about them at the moment, but remember these two receptors, that is MIGM and IgA alpha and Ig beta, are very important receptors of the B lymphocyte. And when a question is asked, B cell receptor complex, you are expected to draw this diagram. You are expected to draw this diagram and explain about these receptors as their function for the activation of the B lymphocyte. Okay, that is as far as B lymphocytes are concerned. So this slide is over. I will now show you the another slide, and that will be on the antibodies. Okay, the antibodies are very important molecules which are responsible for the acquired immune response, and they are the part and parcel of the humoral immunity. Okay, I hope you can see the slide now. So I'll continue with the antibodies. We kept on mentioning about the antibodies which are called as immunoglobulins. Why they are called as globulins? Because they are globular proteins having tertiary structure. Okay, tertiary structure. They are globular proteins. They are folded within themselves. They have beta pleat and certain places alpha helical, and they are folded onto themselves in such a way that they appear like globulins. There are 5 classes of immunoglobulins. Okay, IgG, which is also called as Ig gamma globulin, it is called as, and IgM is called as mu globulin, that is IgM, mu. And IgA is called as alpha globulin, IgD is called as delta, and IgE is called as epsilon. All these are different proteins. Okay, gamma, mu, alpha, delta, and epsilon are different proteins, and they come from the constant region of the antibody gene. They constitute the constant region of the antibody gene. Based on the constant region of the antibody gene, the antibodies are named. Okay, and that constant region recombines with the variable region. Most beautiful thing about the antibody is that it is a hybrid molecule. Okay, it is a hybrid molecule made up of two different chains of proteins coming from two different chromosomes. Okay, one. Second, it is the hybrid of two different genes combined together. So altogether, 4 different genes are there to constitute this protein. 4 different genes are there to constitute this protein. It's a really wonderful molecule. And before the antibody structure was discovered, and before in 1950, Porter and Edelman, Porter and Edelman discovered the structure of the antibody. And they proved that it is a hybrid protein made up of 2 to 3 different types of genes recombined to form a functional protein. Until then, the concept was one gene is responsible for one protein. Okay, one gene, one enzyme, or one gene, one protein concept was there. Beadle and Tatum's concept, it is called as Beadle and Tatum. They had won the Nobel Prize. They had proved that one gene is responsible for one protein, or one gene is responsible for one enzyme. But later, it was proved when the antibody structure was discovered that it has got the components of different genes combined to form a functional protein. Therefore, that one gene, one enzyme concept holds good for few of the genes. Some of the genes will have more than one component. Now, let me come to the point. It is made up of, the antibody is made up of two different chains. The chain that has got largest molecular weight, that is the one which is shown here with the gray color and green color, is called as heavy chain. Heavy chain, which is a largest chain. And the heavy chain, a pair of heavy chain, okay, which is having a molecular weight of 50,000 each. All right, 50,000 each. Then two together, it will be about 1 lakh. And a light chain that is shown in dark orange and light creamish color, the light chain, which is having 25,000 molecular weight. And these two chains, that is a pair of light chains, are identical to each other. Pair of heavy chains are identical to each other. That means they have come from one gene product. Heavy chain has come from one gene product, and you won't believe that it comes from chromosome number 14 in human beings. In chromosome number 14, the gene for heavy chain is present. And the gene for light chain is present on chromosome number 22 and chromosome number 2. Okay, chromosome number 2. The one that is present on chromosome number 2, okay, is the lambda chain. Okay, lambda. And one that is present on chromosome number 22 is the kappa chain. Kappa and lambda. Two different proteins constitute the light chain. Whereas the heavy chain, I already told you that there is the constant region which is labeled as here CH1, CH2, CH3. They are the domains. Constant region of the heavy chain 1, constant region heavy chain 2, constant region heavy chain 3. 1, 2, 3 are the three domains, and this is the typical structure of an antibody. Okay, and this constant region can be up to CH4, where I will tell you a little later. Okay, so the heavy chain comes from chromosome number 14, which is a product of chromosome number 14, and the variable region gene on the chromosome number 14 is present at a far away distance, heavy chain gene, and that recombines with the constant region gene to constitute the heavy chain. Similarly, the variable region gene of the light chain is present far away from the constant region of the light chain. Variable region of the light chain is labeled as VL, constant region labeled as CL. Okay, so these two recombine. So now you know the recombination of these two is somatic recombination, which is taking place in the somatic cell. You had heard about recombination that takes place during meiosis. Remember that. In meiosis, the recombination is seen, and it is in the gametic cells. But these are not gametic cells. This is a strange and a special phenomenon where somatic recombination takes place, where the antibody genes show recombination between two segments of the genes, like CL domain will recombine with VL domain. Same way, CH domain, constant region domain, recombines with VH domain. Okay. So the heavy chain is held together by two disulfide bonds. Minimum is 2, and it can extend up to 11. Okay, it can extend up to 11. So two disulfide bonds are there, and then there are intra-chain disulfide bonds in each of the chains. Intra-chain disulfide bond. These are the intra-chain disulfide bonds, SS, established between the cysteine and cysteine. Okay, intra-chain disulfide bond. And that intra-chain disulfide bond results in the formation of loops like this, loops. And this loop would have approximately about 60 amino acid residues in the loop, the disulfide bond, which is not shown here. You can represent the structure of the antibody with this type of diagram, or you can represent the structure of the antibody like this. And this is generalized structure, which is common for all the 5 classes of the antibodies, with minor changes. And what are those minor changes? I'll come back to it in few minutes. Okay. Now, each antibody molecule having a pair of heavy chain which is held together by two disulfide bonds or more than two, and then the light chain is linked to the heavy chain by a disulfide bond. There is a disulfide bond here. But please note, many a time students make a mistake of showing the bond between these two S and S, which is not there. It is the intra-chain bond, but there is no bond between the S and S here. It is only in this region. Okay. So since it is a protein, it has got NH2 domain, which is becoming NH3 because of the charges, and it has got COH domain on one side of the heavy chain, COH domain on the light chain, NH3 domain of the light chain and CO domain of the light chain, NH3 of the heavy chain, CO of the heavy chain. Okay, so the NH3 domain of the antibody molecule contains the variable region H, that is the heavy chain, variable region light chain, and that constitute the variable region constitute the FAB arm of the antibody, fragment of antigen binding. Antigen binding they have shown here. Okay, fragment of antigen binding. So NH3 domain is the FAB region of the antibody. The CO domain of the heavy chain is the FC region. Okay, so CH2, CH3 constitute the FC region, and that CO domain is responsible for attaching to the membrane. That is the region which attaches to the FC receptor on the membrane of the immune cells. But CO domain of the light chain does not establish contact with the membrane. It is free, but it has got attachment with the heavy chain through disulfide linkage, the Cl domain of the light chain. Okay, so 60% of the molecules, 60% of the time when the antibodies are produced, 60%, it is made up of kappa chain in the light chain region. Okay, 60% of the antibody molecules will contain kappa chain, and approximately 40% of the time, okay, 40% of the time it will be lambda chain. Now, why this discrepancy or why this partiality? We'll see it when we talk about the antibody gene. Okay, we'll talk about the antibody gene. That time you'll come to know why kappa chain is chosen very often compared to the lambda chain. Fine. So each antibody molecule, the variable region domain and constant region domain, if you analyze that, the variable region domain may contain amino acids up to 110. Then it is a repeat like different amino acids, 110 in CH1 domain, 110 in CH2 domain, and 110 in CH3 domain. Okay, so there is a region called as hinge region, hinge, and that hinge region gives flexibility for this arm to bend or turn. It is like a ball and socket mechanism. Okay, ball and socket mechanism. Total number of amino acid residues, total number of amino acid residues in the heavy chain will be about 440 to 446. Okay, 440 to 446, like 110, 110, 110, and some amount of amino acids here, plus 110 amino acids here. It may be less than 110 also sometimes because of the recombination process, and that time it may be 100. Okay, why it becomes 100? I will talk about that when I talk about the immunoglobulin genes. Okay, the loops that are formed for the recombination of the variable region gene to constant region gene, that time some amino acids are going to be eliminated. Okay, eliminated. So that makes the antibody molecule, the heavy chain, having about maximum up to 446 residues. And then the hinge region is included in that. And that region, hinge region, is predominantly made up of proline amino acids, and it is very, very sensitive for papain and pepsin digestion. And that is why sometimes when you have monoclonal antibodies, those monoclonal antibodies will not have FC arm. FC arm. They will have only the FAB because they have been cleaved here with papain or pepsin digestion. And such monoclonal antibodies are called as antibody fragments, fragmented antibodies. They won't have FC region. Now, coming to the important point, why FC region is very important, and if you remove FC region, how it destabilizes the antibody. Let us see. In monoclonal antibodies, since they remove this region to accommodate more number of the FAB regions of the antibody as fragments in a given vial, okay, that antibody is highly unstable and temperature sensitive because this region, FC region of the antibody, which has CH2 domain and CH3 domain, has got biological activity in the sense it can bind to the FC receptor. It can bring about signal transduction. Second, it has got attachment to the sugar residue, which is generally a heteropolysaccharide, and which is attached to the CH2 domain, and that is what is shown here as CHO. CHO is for aldehydic sugar. Okay, it is attached there because the polymer, a short polymer of the carbohydrate is attached. This region becomes highly hygroscopic because glucose and other sugars, galactose, fructose, mannose, which are there as polymers in this region, as the carbohydrate can attract more water. And since water is attracted to this region, the FC region becomes highly hygroscopic. Therefore, when you freeze the antibody below freezing temperature, this region becomes crystallized with the ice deposition. FC region becomes crystallized with ice deposition because of the hygroscopicity. Water molecules accumulate there. Therefore, it got the name FC. F for fragment, C for crystallization. Fragment of crystallization was coined there because that region becomes crystallized with water crystals or ice crystals when it is frozen. Okay, and FAB, you know, it is the fragment of antigen binding. Fine. The CHO represents the sugar residues. Now, when the sugar residues are not there, and when the antibody is not containing the FC arm, that antibody becomes highly unstable. Temperature sensitive, it can get denatured very fast. Therefore, monoclonal antibodies that are prepared without the FC arm are always kept at a below 0 degree temperature to protect them from degradation. Okay, those are the fragment monoclonal antibodies. But those fragmented monoclonal antibodies will not be able to bring about binding to the FC receptor. But all monoclonal antibodies are not like that. Very few. And why? I will talk about that when I'm going to talk about cancer therapy. Okay. So for the time being, keep in mind the immunoglobulin molecule consisting of the heavy chain region and light chain region, in fact, has hinge only in IgG, IgA, and IgD. These three antibodies have the hinge region, and IgM and IgE do not have hinge region. Instead, they have one additional CH domain. So therefore, only IgG molecule, IgA, and IgD molecule made up of three constant region domains: CH1, CH2, CH3. I can call that as C gamma 1, C gamma 2, C gamma 3 for gamma globulin. If I'm using it for the IgA, C alpha 1, C alpha 2, C alpha 3 for IgA. Similarly for antibody D, delta 1, delta 2, delta 3. Okay, so for the IgM molecule, the antibodies will have four domains. CH1 domain here, CH2, CH3, CH4. Either it could be called as CMu1, CMu2, Mu3, Mu4. Similarly, for IgE, it is made up of four constant regions. They don't have hinge region. So since they don't have hinge region, IgM and IgE molecules are considered to be more rigid. Whereas IgG molecule, IgA and IgD, because of the hinge region, they are called as more flexible, because this arm is, now this is shown as a Y shape. It can be shown also as T shape by bending this arm. Okay, T-shaped structure. Antibody can be represented in the form of T shape or Y shape, and this arm is rotatable in IgG, IgA, and Ig delta. Okay, D. But whereas in the case of IgM and IgE, it is not rotatable because it doesn't contain the hinge region. Fine. Then the variable region domain, which is present in the antibody, will have three very important zones, three hyper variable regions in the region here. Okay, variable region domain has three hyper variable regions, and those are very important for the antibody for the specificity. And those hypervariable regions are called as complementarity determining regions, CDRs, complementarity determining regions. Now let us say a particular B lymphocyte recognizes more than 5 antigenic epitopes. Then it will produce 5 different types of IgG molecules recognizing one each for each epitope. Then, when you compare those 5 IgG molecules produced by 5 different B lymphocytes, recognizing 5 different epitopes, that hypervariable region will be different in those 5 IgG molecules. Remaining regions may be same, almost common, but the hypervariable regions will be different. Complementarity determining regions determine the antibody binding capacity with the antigen. And in the variable region, some regions will remain unchanged amongst the different IgG molecules, and they are almost the same amongst the different IgG molecules produced by different B lymphocytes. Then that is called as framework region. Framework region. All right. So that is how the antibodies behave, and their specificity to bind to the antigen lies in the variable region, and that too it is in the hypervariable region or complementarity determining region. That is as far as generalized structure of the antibody is concerned. If you are asked a question to write an account on generalized structure and the structure of the antibody, you may draw this diagram or you may draw this diagram, okay, and explain about it. Thank you.